Welcome and good morning everyone. So today we are going to discuss about the formation of urine. Okay, and as we have known, okay, formation of urine occurs at the nephron, okay, which is in the kidney. Now, what is urine? Okay, urine, okay, is a liquid that contains excess water, right, or surplus of water, salts or mineral ions such as sodium ions, as well as nitrogenous waste products such as urea, uric acid, and creatinine. Okay, now for the SPM, for your SPM level, okay, you must be able to mention that urine contains excess water, salt, and nitrogenous waste product. Tiga benda ni mesti dinyatakan. Right? Now, when it comes to the formation of urine, right, it involves three stages. Okay, the glomerular filtration or ultrafiltration of plasma, which occurs at the glomerular capsule. If you recall, this is the glomerular capsule, or also known as the Bowman's capsule. Right, inside the Bowman's capsule is the glomerular capillaries, and both glomerular capillaries and glomerular capsule, also known as the renal corpuscle or Malpighian body. Okay, so this is where the first stage occurs. Right, the second stage is known as selective tubular reabsorption of needed materials. So meaning to say the glomerular filtrate, right, in the proximal convoluted tubule as well as in the distal convoluted tubule will be reabsorbed, right, reabsorbed into the peritubular capillaries. Okay, so again, let me rephrase. The second stage is known as selective tubular reabsorption of needed materials, which occurs at the convoluted tubules. So when I say convoluted tubules, this second stage, right, occurs at both the proximal convoluted tubules and distal convoluted tubules, right? And then the third stage is tubular secretion, right? Tubular secretion at the convoluted tubules. Again, I mentioned convoluted tubules. So this tubular secretion occurs at the proximal convoluted tubules and at the distal convoluted tubules. Okay. Now this ultrafiltration of plasma, okay, is a passive process. Okay, where hydrostatic pressure, right, from the afferent arteriole, right, will force small molecules like water, glucose, and urea out from the blood plasma in the glomerular capillaries into the Bowman's capsule. Okay, so I need to say, you know that afferent arteriole, okay, brings blood, okay, which has nutrients, okay, into these glomerular capillaries. Because of the high hydrostatic pressure of this afferent arteriole, right, this will push the content, right, of the blood in the glomerular capillaries into the Bowman's capsule or glomerular capsule. Right. Another factor contributing to this hydrostatic pressure, right, high hydrostatic pressure, is the larger diameter or wider diameter of the afferent arteriole compared with the efferent arteriole. Okay, ini besar ni diameter dia ni. So tekanan di sini mesti kuat, sini pula tekanan rendah. Belah sini tekanan kuat. What happens to the content in the glomerular capillaries? Obviously, the contents, right, will be pushed out from the glomerular capillaries into the glomerular capsule, and this content is known as glomerular filtrate. So this glomerular filtrate will enter, okay, into the renal tubule. Okay, renal tubule. This is the renal tubule, or also known as the proximal convoluted tubule. So in this proximal convoluted tubule, right, as well as the distal convoluted tubule, selective tubular reabsorption occurs, right? Okay, so to be more specific, reabsorption of useful substances, right, like glucose, amino acids, hormones, from the tubules to the blood capillaries occurs. This blood capillaries here is known as the peritubular capillary. Okay, now as the filtrate passes along or travels along the tubules, tubular secretion occurs. Okay, with unwanted substances like toxin, drugs, ammonia. Remember, ammonia is poisonous to our body. Okay, excess potassium ions and hydrogen ions will be secreted into the renal tubule from the peritubular capillary. Okay, okay, now class, this diagram here shows you, okay, how the cells, right, how the tubular cells, okay, look like, okay, from one region to another region in the nephron. Okay, so in the glomerular capillaries, you can see this is the glomerular capillaries, right, and this is the podocytes, okay, which has this like finger-like projections that function as a filtration slit. So because of this, okay, the content from the glomerular capillaries able to be filtered out to form the glomerular filtrate. Okay, now we have these proximal convoluted tubule cells or tubular cells, okay, that present at the proximal convoluted tubule, and this is the tubular cells that present at the distal convoluted tubules. Apparently both cells, okay, are very look alike. Okay, both cells apparently they are the simple cuboidal epithelial cell. If you look closely, both are simple cuboidal epithelial cell. Okay, except proximal convoluted tubule cells, they have microvilli. You nampak, and the mitochondria are abundant compared with the distal convoluted tubule cells. Okay, then if we compare with the loop of Henle and this thin segment, this is the loop of Henle. Loop of Henle has two regions, right? The thick segment and the thin segment. Okay, this is the descending, this is the ascending arm. Descending arm has the thick segment and thin segment. Ascending arm has the thin segment and the thick segment. Okay, so apparently the cells are simple squamous epithelium cell. Okay, for the loop of Henle, the thin segment. For the collecting duct, all right, they consist of simple cuboidal epithelial cell. Okay, with combination, right, you can see some lacks of microvilli, some has microvilli. Okay, the ones with the microvilli are also known as intercalated cell. Okay, the ones without the microvilli are known as the principal cell. Okay, so to STPM, you must be able to differentiate, okay, how the renal tubule cells look like, okay, how they are differ from each other. All right, now let's discuss um, into more specific, right, at each of those three stages. So the first stage is glomerular filtration. Now, this glomerular filtration, like what I mentioned earlier, all right, takes place in the glomerulus. Okay, if you recall, this glomerulus, okay, is the network of capillary. You can see this network of capillary, okay, that is surrounded by the Bowman's capsule. All right, this is the Bowman's capsule, and this is the Bowman's space, to be more specific. Okay. Now, this glomerular filtration is a non-selective process, okay, with regard to small molecules. Meaning to say, this glomerular filtration only allow small molecules to pass through or to be filtered out from the um, glomerular capillaries into the Bowman's capsule or Bowman's space. So apparently both useful and waste substances are filtered into the Bowman space from the glomerular capillary. Okay. This process contributes to homeostasis. How? Because this process removes toxin or waste, okay, product from the blood. Now, this is the afferent arteriole. Okay, this afferent arteriole, as you have known, like what I mentioned earlier, blood that enters or came from the afferent arterioles, right? They are in high hydrostatic pressure. Okay, so blood enters the glomerulus, this glomerulus, okay, at high hydrostatic pressure from the afferent arteriole, okay, because of the high hydrostatic pressure blood, right, from the renal artery. And then the afferent arteriole, the diameter of the afferent arteriole is wider than the efferent arteriole. So this again create high hydrostatic pressure in the glomerulus that forces all the substances except blood and plasma proteins through the pores of the capillary wall of the glomerulus into the Bowman's capsule, right? The highly coiled, okay, you can see this glomerular capillary is highly coiled, okay, and great permeability walls of the glomerular capillaries and filtration slits. These are the filtration slits. You can see it's here, okay, and the filtration slits between podocytes serve as a large surface of filtration membrane. Okay, so let me rephrase that. The highly coiled, okay, this glomerular capillary is highly coiled, and then the walls are highly permeable. Okay, walls are highly permeable to substances to pass through, and then the existence of filtration slits between podocytes. These three conditions, okay, made these glomerular capillaries serve as a large surface of filtration membrane. And then the high hydrostatic pressure, right, that I mentioned earlier, okay, will force the removal of substances such as water, glucose, and urea, right, except blood plasma to form glomerular filtrate that passes the filtration slit into the Bowman's space or capsule space. So now the content that we have here is known as glomerular filtrate, which has nutrients like glucose, okay, and then waste products such as urea or any toxins, right, and excess water, okay. Those are the content of the glomerular filtrate. Now, glomerular filtrate does not have, okay, blood and plasma protein. Now, talking about these podocytes, right? These podocytes, they are specialized epithelial cells, okay, specialized epithelial cells that make up the inner wall of the Bowman's capsule. And then these podocytes, they form very porous slit-like network, okay, slit-like network that surround the glomerulus to assist in the filtration. Now another cell is, right, these juxtaglomerular cells. Can you see? It's here, right? These juxtaglomerular cells, okay, also known as granular cells. These are specialized smooth muscle cells, right? Specialized smooth muscle cells in the walls of the afferent arterioles that deliver blood to the glomerulus. Remember afferent arterioles? Okay, this afferent arteriole deliver blood to the glomerulus, and the blood contains toxin as well as nutrients. Now, these juxtaglomerular cells, right, synthesize, store, and secrete renin. Renin is an enzyme, okay, and this renin is needed to regulate, right, to regulate the blood pH and blood volume via the renin angiotensin aldosterone system, which we learn in the next video. Okay, so this is the glomerular filtration process. Okay, the first stage in the formation of urine. Okay, so this is the podocytes. Okay, this is their sort of like feet. Okay, which gives or form the filtration slit. Can you see this filtration slit? The purple region here. These are the filtration slit. All this purple region. Okay, so in this purple structure here apparently is the glomerular capillaries. Okay, the empty space here is the Bowman's space. Okay, now selective tubular reabsorption of needed materials. So reabsorption of useful substances, right? So because remember earlier during the ultrafiltration, what happened? Even nutrients are being filtered into the Bowman's space, which form the glomerular filtration. So in the glomerular filtration, right? In the glomerular filtration here, okay, in the glomerular filtration we have nutrients, and those nutrients must be absorbed by our body. So the absorption of those nutrients occurs here at the selective tubular reabsorption of needed materials. So reabsorption of useful substances like glucose, vitamins, amino acids, and hormones from the renal tubules to the blood capillaries, okay, peritubular capillaries, but leaves waste and excess of other substance like salt to be excreted in the urine. So this contributes to homeostasis. So let me rephrase. The glomerular filtrate that passes through this renal tubule, right? Any nutrients or useful substances from the glomerular filtrate will be reabsorbed, okay, from the glomerular filtrate in the renal tubule. Unwanted substances, right? Unwanted substances such as urea or excess substances like salt will remain in this renal tubule. Okay. So the glucose, amino acids, sodium ions, potassium ions are partially reabsorbed by active transport, okay, in the renal tubule, or to be more specific, the proximal convoluted tubule and the distal convoluted tubule. Okay, you can see here sodium ions, potassium ions, chloride ions as well. Okay, so they are being reabsorbed, and sodium ions, potassium ions, amino acids, and glucose are reabsorbed by active transport, okay, against the concentration gradient, while other sodium ions, potassium ions, bicarbonate ions, and chloride ions are being reabsorbed by passive transport, okay, via channel protein or protein or co-transport protein. So let me rephrase. Okay, reabsorption of water is by osmosis, simple diffusion osmosis. Bicarbonate ions by passive transport, okay, which involve pore protein, channel protein, or co-transport protein. Okay, reabsorption of sodium ions are by active transport. Some are by passive transport as well. Okay, chloride ions by passive transport. Okay, this selective tubular reabsorption is highly selective. Okay, this process is highly selective, and 65% occurs in the proximal convoluted tubule. So the other 35% occurs at the distal convoluted tubules. So 99% of the glomerular filtrate is reabsorbed back into the blood, okay, via the peritubular capillaries. Mostly water is reabsorbed here, okay, via osmosis. Now, like what I mentioned beforehand, the tubular cells are simple cuboidal epithelial cells, okay, that lining the renal tubule. And these tubular cells are adapted for reabsorbing the glomerular filtrate, okay, by their abundant microvilli that increase the surface area for tubular reabsorption. Okay, and then these tubular cells, okay, contains numerous or many mitochondria. Okay, these mitochondria provide energy to running the cell pumps that actively transport nutrients and materials to the peritubular capillaries. So meaning to say, these mitochondria are needed because these mitochondria provide ATP for the active transport. Okay, the active transport involves sodium potassium pump. This pump needs ATP. So the ATP comes from the mitochondria. And then these tubular cells has many pinocytic vesicles. You can see it's here. Many pinocytic vesicles that carry out pinocytosis of the glomerular filtrate into the tubular cell. Okay, pinocytosis is drinking of the liquid. So this tubular reabsorption continues as the glomerular filtrate passes through the loop of Henle. This is the loop of Henle, okay, and the distal converting tubules. The filtrate is further concentrated as it passes through the collecting duct that leads to the renal pelvis. Okay, so the content of the glomerular filtrate starts from the renal corpuscle all the way to the renal tubule. Okay, as the glomerular filtrate enters the collecting duct, the concentration, right, of the filtrate or glomerular filtrate is higher. Okay, masa sini dia cair, sampai di sini collecting duct dia jadi pekat dah. glomerular filtrator. Okay, so this is what I mentioned, what I meant earlier. Okay, how the glomerular filtrate here is being reabsorbed selectively. Okay, through the um, renal tubular cells into the peritubular capillaries. So daripada sini masuk dalam sini ke blood capillary atau peritubular capillary. Okay. Paracellular path, transcellular path. Paracellular path, um, the content, right? The glomerular filtrate are reabsorbed by passive transport through the sites of the cell. Transcellular, the content are reabsorbed through the tubular cells and then towards the peritubular capillary. Okay, you can see the sodium potassium pump here. Okay, now paracellular path normally water just by simple diffusion from the lumen of the tubular cells, lumen of the renal tubule. Okay, simple diffusion towards the peritubular capillaries. Solutes, salts are by active transport. Okay, sorry, solutes, some by active, some by passive transport. Okay, again, this slide here to show you how the peritubular capillaries, how the proximal convoluted tubule, right, the tubular cells in the proximal convoluted tubules look like compared with the tubular cells in the distal convoluted tubule. Okay, so students, you must be able to differentiate how these two cells look like. Okay, tubular cells at the proximal tubule, tubular cells at the distal tubules. Now, tubular secretion. So this tubular secretion is an active process where only certain ions like potassium, hydrogen, ammonium ions, right, and then toxin and drugs are secreted from the peritubular capillaries into the renal tubule. Right, so in tubular secretion, what happens? The content from the peritubular capillaries are secreted into the renal tubule. It's the other way around compared with the reabsorption. So this helps to remove waste and unwanted substances from the blood, as well as to regulate the pH of the blood by the controlled secretion of hydrogen ions and reabsorption of the carbonate ions. This contributes to homeostasis. Now, please take note that the secretion ions and other substances are moved across the tubule epithelium in the opposite direction of the tubular reabsorption. So mainly this tubular secretion occurs in the region of the distal convoluted tubules. Okay, so we have like this is where the peritubular capillaries. Okay, so the content of the peritubular capillaries, like the potassium and hydrogen ions, are being secreted into the renal tubule. That's why we have this arrow from this direction. This represents the tubular secretion, and this arrow here represents the tubular reabsorption that we have discussed earlier. Okay, production of concentrated urine depends on a high salt and urea concentration in the interstitial fluid of the kidney medulla. Salt is most concentrated at the bottom of the loop of Henle. Right? I will explain this in the countercurrent mechanism later. Urine is a watery solution of nitrogenous waste. Excess salt, okay, excess water, and other unneeded substances. So we already covered the three processes that involved in the formation of urine. Now, when it comes to explaining formation of urine, Okay, apart from these three processes, you must include the counter current mechanism as well, which explain what happens at the loop of Henle, or how concentrated urine is being produced. Okay. Now, this counter current mechanism is a mechanism that causes the thin descending loop of Henle permeable only to water. Okay, permeable only to water, but impermeable ni tidak membenarkan. Okay, impermeable to sodium ions and urea, while the thick ascending loop of Henle, okay, permeable to salt, which is the sodium ion and chloride ions, but less permeable to water. Okay, this will cause the medulla, to have low water potential. Okay, and then the urine to be concentrated, and more water can be reabsorbed to the vasa recta to maintain homeostasis. Okay, when more water is absorbed back to the vasa recta, meaning to say our body will receive more water. Okay, if less water is reabsorbed into the vasa recta, the water will be excreted in the urine. Okay, so we don't want that to happen. Okay, our kidney, it wants to conserve the water for our body, keep the water for our body. So desert animals have more juxtamedullary nephron, okay, for more water absorption. If you recall, juxtamedullary nephron, it has longer loop of Henle. When you have longer loop of Henle, recall I mentioned that the thin descending arm loop of Henle is very permeable to water. Okay, so meaning to say more water will be reabsorbed back by the body. So the loop of Henle is specialized to highly concentrate sodium chloride in the interstitial fluids, right? Interstitial fluids of the medulla, which causes the kidney to produce concentrated urine. So now in the countercurrent mechanism, first the glomerular filtrate become more concentrated as water diffuses, right, via osmosis into the vasa recta from the descending loop of Henle. Can you get that? The water from the glomerular filtrate, as it passes down in the descending loop of Henle, this water will diffuses via osmosis. Okay, the water will diffuses via osmosis out from the descending loop of Henle. So the water will enter the vasa recta. Will diffuse into the vasa recta. So the glomerular filtrate loses water. So what happens to the glomerular filtrate? It becomes more concentrated. Next, diffusion of sodium chloride ion in the ascending loop of Henle contributes to high sodium chloride concentration of the interstitial fluid in the medulla surrounding the loop of Henle. Now, in the descending, the thin descending loop of Henle, water diffuses out by osmosis, right? In the ascending thin loop of Henle, right, sodium chloride will diffuse out from the renal tubule, okay, or the sodium chloride ion will diffuse out from the thin ascending loop of Henle into the medulla, right, into the medulla or into the interstitial fluid in the medulla. Okay, sodium chloride ions will diffuse out. Now, why did I stress out diffuse here? Because this is a passive transport. Why sodium chloride ion can diffuse by passive transport in the thin ascending loop of Henle. Why? Because the concentration of sodium chloride ions here is very high or higher. Okay, it's very high. Why does the concentration of the sodium chloride ion here is very high? Because water already diffused out from the glomerular filtrate in the thin descending loop of Henle. So the concentration of solute here, which is sodium chloride ion, is very high or higher. So the sodium chloride ions just diffuse, okay, out from the ascending arm or ascending loop of Henle, follows concentration gradient. Okay. Now, in the thick ascending loop of Henle, right, or the thick ascending arm loop of Henle, the movement of the sodium chloride ions are not by passive transport, it is by active transport, because the concentration of the sodium chloride is lower. Okay, how does it become lower? Because some of the sodium chloride ions already diffuse here in the thin ascending loop of Henle. So the ones that left here is only few. So the remaining sodium chloride ion have to move by active transport out from the thick ascending arm loop of Henle into the medulla. And the movement of these sodium chloride ions are against the concentration gradient. Okay, now the glomerular filtrate in the distal converting tubule is diluted. Okay, is diluted. Why? Because sodium chloride ions, right? Sodium ions and chloride ions, or sodium chloride, is actively transported out to the cortex. Okay. Now, always remember, sodium chloride. When it comes to sodium chloride, right? The sodium is transported actively. Okay, the sodium ion is transported actively. After the sodium ion is already transported actively, the chloride ion will follow passively via co-transport protein. Okay, so sodium chloride also actively transported out from the collecting duct. You can see here. Okay. Class, please do not confuse on the transportation of the sodium chloride. In the active process, right, when the concentration of the sodium chloride is high, right, in the concentration of sodium chloride is high, the sodium chloride will just diffuse. Both sodium ion and chloride ions follows the concentration gradient. But when the concentration of the sodium chloride is lower, right, the sodium ion will be transported by active transport, and then the chloride ion will follow passively, okay, via the co-transport protein. Remember, during your first sem, you learned about co-transport protein. Now, the hypertonic interstitial fluid. Remember, all these sodium ions, chloride ions, they are transported into the medulla, right? To the medulla. Okay. So, what happened to the interstitial fluid in the medulla? The interstitial fluid in the medulla become hypertonic, right? You need to say this interstitial fluid has high sodium content but low water potential. Okay, so this will draw water osmotically from the filtrates in the collecting duct. So meaning to say, in the collecting duct, more water from the filtrate will be diffused, right, by osmosis into the medulla. This is very possible because of the hypertonic interstitial fluid here. Okay. Now, this collecting duct, they are permeable to urea. Okay, so some urea will diffuse out from the collecting duct. So this contributes to the high solid concentration in the inner medulla. This process helps to concentrate the urine. Okay, so hypertonic urine conserves water. When this collecting duct being permeable to urea, right, the urea diffuse out into the inner medulla. What happened to the interstitial fluid here? Yes, the interstitial fluid here become hypertonic interstitial fluid. So what happens to the remaining water in the glomerular filtrate? The remaining water will diffuse out from the collecting duct into the hypertonic interstitial fluids. Okay, so the concentrated solutes, right, any remaining sodium chloride as well as urea, okay, and toxins and any unwanted substances will be excreted in the urine. Okay, so that's about it on the formation of urine. Now, osmolarity is the measure of solid concentration on different regions or in the kidney. So now, if you recall, the tip loop of Henle has the highest solid concentration. Okay, it has the highest solid concentration or the highest osmolarity. Why? Why this bottom, sorry, not tip, bottom, why does the bottom loop of Henle has the highest osmolarity or highest solid concentration? Because in the thin descending loop of Henle or thin descending arm loop of Henle, water already diffused by osmosis. So when water already diffused, what's left in the glomerular filtrate here? Mostly the solutes. So the osmolarity here is very high, right? Then as we go up in the ascending loop of Henle, what happens? In the thin ascending loop of Henle, some sodium chloride diffuse out. In the thick region, ascending loop of Henle, some sodium chloride are being actively transported out. Remember I mentioned earlier, this is where the active transport of sodium ions occur, and this is where the passive transport of the sodium chloride ions occur. Okay, again, this is to show you how these peritubular capillaries, okay, look like. Okay, and then this is how the proximal look with the many microvilli. This is the distal, okay, legs of microvilli. Okay, and then this is the cross section of the loop of Henle. Okay, so this is the ascending limb, this is the descending limb, this is the thin descending limb. All right, this is the thick descending limb. So these are the capillaries, this is the collecting duct. All right, so that's it for today. Thank you for watching and happy studying.